Okay, so in this handout we're going to be demonstrating bacterial cell replication. Basically, replication is the process by where a cell will duplicate its DNA. With bacterial cells, they like to duplicate their DNA obviously when they're going through cell division. In bacterial cells, we call this binary fission, but basically the bacterial cell wants to produce another daughter bacterial cell. And in order to make that happen, what first needs to be done is the DNA needs to be duplicated for the daughter cell. And this is a process that we know as replication. So replication in a prokaryotic cell is going to happen a little bit differently than what it would normally do in a eukaryotic cell. With prokaryotic DNA, it's not found inside of a membrane-bound nucleus, so basically replication is going to occur in the cytoplasm. And also the chromosomes of prokaryotic cells look different than what they would in eukaryotic cells. Eukaryotic cells typically will have linear chromosomes, and they will typically have more than one. Our cells have 46. With prokaryotic chromosomes, typically they are just going to have one chromosome, and it is going to be a circular chromosome. So in order for replication to begin, the first thing that needs to happen is we need to basically unwind the double-stranded DNA. DNA is held in kind of a helical format, so in order to allow the 30 different enzymes that are responsible for replication of DNA access to the DNA, it needs to be unwound and basically unzipped. There are specific enzymes that are going to unwind the double-stranded DNA called topoisomerases. A really important enzyme that does come in and separates the double-stranded DNA is called DNA helicase enzyme that we see here. So basically, with this DNA being double-stranded, the DNA helicase enzyme is going to come in and break the hydrogen bonds that hold the nitrogenous bases together. So this always starts at a region called the origin of replication. This is typically an AT-rich region, and we see that this would have been this region right here. These are two separate DNA strands at this point that had already been unzipped. But this AT-rich region allows us to unzip the DNA using less energy. The nitrogenous bases are held together by hydrogen bonds. The A to T association has 2 hydrogen bonds, where the G to C association has 3 hydrogen bonds. So obviously it's going to take less energy to break 2 hydrogen bonds versus 3 hydrogen bonds. But the really important thing here is that we want to separate this double-stranded DNA molecule into 2 single strands so that all our replication machinery can come in and synthesize new strains. And again, that is done with the helicase enzyme. Now, once these 2 single strands are separated, they automatically want to go back together again. Based on the rules of complementary base pairing, those A's are going to want to go back with the T's, and the G's are going to want to go back with the C's. But we need to keep them separated so our replication machinery can get in there. And how we do that is using something called single-stranded DNA binding proteins. You'll see these as these little dots here. These are little proteins that are going to bind to each of the single strands of DNA and keep them apart so they don't automatically go back together again. That way they're held apart so the replication machinery can come in and we can start synthesizing the new DNA. Okay, so now that our 2 single strands are being held apart, we are ready to start actually replicating DNA. So the main enzyme that's actually responsible for reading the code and adding the corresponding nucleotides is DNA polymerase III. Now DNA polymerase III is kind of tricky in the way it works. One way that it can work is it only will add DNA nucleotides to an already formed 3' end. So we actually already have to have something laid down here for DNA polymerase III to come in and work off of. It can't just go in and start adding DNA right away. It needs to build off of something. So that is where RNA primase comes into play. RNA primase is an enzyme that's going to come in and it's going to lay down a short RNA primer. This is kind of going to serve as a starting point for where DNA polymerase III can come in and build off, because remember, it can't start off of nothing. So the RNA primase is just going to serve as exactly what it says. It's a primer. So here we're going to be starting, this is our origin of replication. So the RNA primase is going to come in and read this AAT code. Now to this it is going to add a complementary RNA primer. Now remember, with RNA we don't see thymine, we actually replace it with uracil. So that this code says AAT, our RNA primer is going to read UUA. At this point we have a short RNA primer, and DNA polymerase III can come in and actually start reading the rest of the code and add to that growing 3' end that we see here on the RNA primer. So DNA polymerase III is going to come in, it's going to read a C and it'll add a G, it'll read a C and add a G, read a G and add a C, read a C, add a G, G, C, A, T, T, A, C, G, and it will keep working in this direction, in this 5' to 3' direction, until it gets to the end. Another thing with DNA polymerase III is that it only works in the 5' to 3' direction. So this strand that we're reading here is in the 3' to 5' direction. So when we make the complementary base paired, we're going to be making it in the 5' to 3' direction, which is the exact direction that the DNA polymerase III needs to work in. So it's going to be able to go through all the way down to the end, and because it can do this without stopping, we call this continuous. So here, this strand that we're synthesizing is called the leading strand, because the leading strand is synthesized continuously without stopping. And that has to do with the fact that DNA polymerase only works in that 5' to 3' direction. And that is the actual direction that we are working here. We are working in a 5' to 3' direction. Okay, so when we start copying the other template strand of DNA up here, we actually run into some complications because of the directionality and how RNA polymerase III works. So if you look up here, the template strand is running in a 5' to 3' direction, but we always make the new strand anti-parallel to that. So theoretically, this strand would need to be made in the 3' to 5' direction. So we have a growing 5' end here if we were going this way. But remember, DNA polymerase III can only add to a growing 3' end. So this growing 5' end here is basically useless. We cannot work this way on the other strand of DNA. So what needs to happen is we actually have to work backwards here, which makes this a little bit more complicated. So the first thing that's going to happen is RNA primase is going to come in again, and it's going to lay down a series of short RNA primers. And we see those a few right here, and then there's one right here. Basically, these short RNA primers are going to be laid down in a 5' to 3' direction. So we have a G here, we add a C, we have an A here, we add a U. What we have just made is this little RNA primer running in 5' to 3' direction. We do the same exact thing up here. We have a C, we add a G, we have an A, we add a U. We created another RNA primer running in the 5' to 3' direction. What we're doing is we're creating these little 3' ends here. So here will be a 3' end, here will be a 3' end. That way, DNA polymerase III can come in and add to those growing 3' ends. And what we're doing is we're synthesizing this new DNA template, or this new DNA strand, in the opposite direction that the helicase is running. When this happens, we're basically creating short segments. These short segments are called Okazaki fragments, and that's because the DNA polymerase III can only add nucleotides in a growing 3' end, so it has to work backwards in short segments off of these little RNA primers, and that's what gives us our Okazaki fragments that we are talking about. Now, because this strand is produced and leads to Okazaki fragments, we call the synthesis of this strand discontinuous, and it is referred to as the lagging strand. Now obviously these little fragments don't exist once the replication has been completely terminated, so these are kind of glued together eventually to form kind of a continuous strand, and that is actually done using the enzyme called ligase. Again, ligase basically acts like a glue and kind of glues these Okazaki fragments together, because obviously you just don't have segments of DNA existing in your chromosomes. Another thing that you don't have randomly existing in your chromosomes, or bacterial cells for that matter, is they don't have random bits of RNA inside of their DNA chromosomes. So we do need to remove all these RNA primers that we have put in here throughout the whole process. The enzyme that does that is called DNA polymerase I. So DNA polymerase I is going to remove all RNA primers on both the leading strand and the lagging strand, and replace them with DNA, because we just don't want random sections of RNA held there forever. So it is replaced, excised and replaced with DNA polymerase I. So at the end of replication, you have just taken this double-stranded DNA molecule, you separated it into two single strands and made two daughter strands of DNA. This is done in a semi-conservative pattern. So basically, the two new daughter strands are part old and part new, and we saw that. Basically, the part old comes from the parent strand that we were using as a template, and the part new comes from the strand that we were just synthesizing. So even though the leading and lagging strands are synthesized quite differently, at the end they look exactly the same when we get two new daughter strands that are semi-conservative. They are part brand new DNA and part old DNA that was used from the original parent strand to serve as a template.